Hello and welcome to today's PIR live event webinar brought to you by Partners in Research Canada. My name is Stacey Joyce and I will be your host. Don't forget that you can use the chat function with the button near the top left of your screen to ask questions throughout the webinar, and we will relay them to our guest expert on your behalf during the Q&A portion. Please don't forget, while you are writing your question, to select everyone in the to field so that everyone who's participating today can see all of the questions being asked. So today's guest is Sangeetha Paramathas. She is a PhD candidate in biophysics at the University of Toronto. She's a researcher at Sick Kids Hospital, and she's a member of the Canadian Cancer Society's Research Information Outreach team called R.I.O.T. Thanks so much for joining us. I'll let you take it away.
Perfect. Okay, does that look okay?
It does indeed.
Awesome. Okay, thank you so much for having me speak today on in preparation for World Cancer Day. And thank you so much, Stacey, for the introduction. So my name is Sangeetha, and I'm going to speak to you a little bit about cancer and cancer research. So I am representing the Research Information Outreach team called R.I.O.T at the Canadian Cancer Society. And who we are, we are a group of volunteers. Most of us are graduate students at master's or PhD level. Some of us have already graduated, and some of us are still doing research at the postdoctorate level. And what we want to do is share our knowledge and love for research, particularly in the field of cancer, and highlight current research advances and share that with the general public. We post blogs and do presentations similar to the one I'm going to talk to you about right now to classes, classrooms, and students across Toronto. Okay, so I'm just going to dive right into it. So a lot of what we understand about cancer really depends on what we understand about normal cells. So you guys are probably already familiar with the concept that human tissue or animal tissue is composed of millions and millions of cells. And these cells are organized in an organized and structured manner in the form of tissue. Here's a picture of a typical skin tissue in humans, and you can see the very organized structure found in the human tissue. You have different layers, and each layer is composed of different types of cells, and they have different functions. The cells have to know when to start dividing and when to stop dividing in order to maintain this function and this very organized structure. These cells also have to be able to respond to signals from the environment in order to do their function properly. Oftentimes in cancer, cancerous cells don't know when to stop dividing and thus aren't able to maintain proper structure and function. When we talk about cells, we like to compare it to a city. So we say a cell is kind of like a city. In a city, you have multiple different structures, multiple different people, and they all have specific functions. Just like in a city, if a specific person, like a doctor or an engineer with a specific function, is unable to work, or if a specific structure, like the roads, aren't able to function, then you have a city that might be in chaos. Similarly, in a cell, the structures and the particular functional units in the cell have to act and develop properly in order for the cell to remain healthy. In the city, the functions is probably carried out by multiple different people, and they have unique roles. In the cell, the functional unit is what we call proteins, and every protein has a specific unit, a specific function that is integral for the cell's healthy living. The protein might be involved in cell division, it might be involved in the cell's movement. So in cancer, we understand that cancer is a disease of abnormal cells, and that a lot of the abnormal changes that occur in cancer cells are often due to the genes and proteins. So as I mentioned in the previous slide, if a protein is not functioning properly in a cell, then the cell becomes abnormal. I'll talk a bit about what genes are in the next slide. And we know that cancer can occur in many different parts of our body. And to date, we understand that there are over 200 different types of cancers. And oftentimes, each of these cancers can be attributed to different abnormal functioning proteins or genes. Okay, so we spoke a lot about genes and proteins, but where do they actually come from? You are probably familiar with the concept of DNA. So DNA, as you know, is the information inside your cell that makes you who you are, and it tells the cell to become a cell and what kind of cell. So in characteristics that make you you, like your hair color or your eye color, are attributed to specific information in the DNA that produces proteins that give you that characteristic. You can kind of think of DNA like a cookbook. So the cookbook has all the different recipes and all the different information to make a variety of different foods and food dishes. The DNA also has all these different informations to produce different qualities of a cell and different qualities of tissues that make you you. But if you kind of go further into it, you know that a cookbook is made up of many different recipes. Recipes for pastas, recipes for desserts. Just like that, the DNA is also packaged into smaller units called genes. So each gene has the instructions to make something specific, just like a recipe has the instructions to make something specific of a food. A recipe obviously produces a food dish, whereas a gene has the instructions to make proteins. So in a normal cell, the instructions in the gene are all correct, and that produces a normal protein. But oftentimes in a cancer cell, the instructions might be incorrect, and we like to call that a mutation. So if the instructions are incorrect, then that will produce an abnormal protein. Sometimes the protein might function not properly, and other times it might not function at all. And this is inherent to what produces a cancerous cell. Something in the gene of that cell that produces an abnormal product that leads to cancerous cells. So we understand that normal cells are able to divide and know when to stop dividing, whereas cancer cells are also able to divide, but they don't know when to stop dividing. Normal cells can sense the environment and can respond to the environment, whereas cancer cells may not be able to sense the environment or may choose to ignore the signals that come from the environment. Normal cells always talk to the environment and talk to other tissues through signals, whereas cancer cells don't or may do that incorrectly. Normal cells, if they become sick, are able to self-destruct so that they can, not produce any bad functions in the tissue that they're found in. And we like to call that apoptosis. Whereas cancerous cells, even if they're sickly, they don't self-destruct. Instead, they continue to divide and continue to produce improper function in that tissue. All cancer types have certain characteristics that make them cancerous, and we like to call this the hallmarks of cancer. Some of these include the cancer cell's ability to avoid the immune system. So oftentimes when your immune system sees that there's something wrong with a cell or sees an abnormal cell in your body, it can go in and detect that cell and destruct it, whereas cancer cells often avoid that. Cancer cells can often move around in the body, and we call that metastases. If you have a cancer in your lung, it might be able to move to another part of your body. Cancer cells, because they're always dividing, they need a lot of nutrition, and cancer cells are able to hijack the production of blood vessels to feed themselves and self-sustain them to help them grow. As we spoke, cancer cells have a lot of genes that are incorrect or have coding errors in them, and thus produce a lot of mutated genes that produce abnormal proteins. Lastly, we talked about how cancer cells can resist death, so they continue dividing even when they're not supposed to. So as cancer researchers, we kind of act like detectives. We hope to find clues that can help us either prevent or treat cancer. A lot of different cancer researchers work in different fields of cancer. Some of us are working in areas that try to look at ways to prevent cancers. Others look at ways to detect cancer, and I'll speak a little bit about this because that's where my research interests lie. other cancer researchers often work in the areas of treating cancer. So what kind of drugs can we develop to treat cancer? What's going to work? What's not going to work? And lastly, there are other research that are looking at ways to help cancer survivors lead a healthy life. And this is called survivorship research. So my research, I work in the laboratory of Dr. David Malkin. We're located at the Hospital for Sick Children, and our research, or my research interest, is on how to detect cancer early on. So I'm going to speak a little bit about this. We know that there's something cool that happens to cells when they die. When cells die, they are able to shed their DNA into the blood. So what do we know about normal cell DNA and cancer cell DNA? So we know that cancer cell DNA is often mutated. So there's a lot of coding errors in the DNA, whereas normal cell DNA is mostly unmutated, so it's normal. So if a normal cell dies and releases its DNA into the blood, and a cancerous cell dies and releases its DNA into the blood, if we take a sample of a patient's blood and look at the DNA found in that blood, in a healthy individual, the blood will always contain DNA that is unmutated. Whereas in a cancer patient, because they have a tumor growing in their body and the tumor cells might be dying, the tumor cell's DNA will be found in the blood and it will be mutated. So we can look at the DNA in the blood and find and detect whether the DNA in that person is mutated or unmutated. And that can be an indication of whether that person has cancer and what stage of cancer they have. And this technique can be applied to a variety of different cancer types. Some cancer types that you probably are familiar with include breast cancer, lung cancer, colorectal cancer. All these cancer types can produce tumor cells that die and release their DNA into the blood. So we can take a sample of the patient's blood and look at the DNA content and say, hey, it has these mutations, so it must be this cancer. And how can we use this? We can use this technique for early detection. If we can detect the cancer early in a patient, then we can treat them early on and they'll have good disease prognosis. We can also use this technique to monitor the tumor. Is the tumor shrinking? Is it growing? Has the tumor come back? We can, if a patient is undergoing treatment, we can use this technique to say if the patient is responding well to the treatment or is not responding well to the treatment. And lastly, we can look at this DNA to see how it's changing over time, to see if the tumor is evolving. Because oftentimes the tumor is changing and growing in the person. And this is a good way to see what stage the tumor is at. That's kind of a little bit about what my research focuses on. And we're particularly interested in early detection because we work with childhood cancers. So I'm actually going to end there. And thank you for listening to me. Hopefully that wasn't too confusing. So if you are interested in keeping up with cancer research, I highly encourage you to follow us on our website at toronto.riot.com or on Facebook and Twitter, because we post a lot of videos and blogs that summarizes the current research advances in cancer that you might be interested in. Thank you.
Thank you so much for that presentation. I certainly learned a lot. And I have watched the lab tour videos for the R.I.O.T team. They are quite good and nice and concise. So if you haven't had a chance yet, you can search for those on YouTube and have a look at what it looks like where Sangeetha does her research, or at least a similar lab, and the similar instrumentation and fridges and dark rooms and lots of good stuff that they
all
do.
the typical lab stuff.
So if you want to go ahead and hit the stop share button, Sangeetha, then we'll be able to see your picture a little bit larger. Excellent. While we're answering questions. First off, just a question about your research, since that's where we left off, a little more specific on the blood testing that you do. How can you tell what is mutated and what is unmutated DNA in the blood samples?
So the DNA is made up of 4 types of molecular units. They're A, T, G, and C. So depending on what order and what combination they are in the DNA, that combination codes for the gene. So if those 4 units are found in incorrect places or in an incorrect order, we call that a mutation. So if we compare a normal gene to a mutated gene, we can see how, in what order these units are aligned. And if they're not aligned correctly, then we know what the mutation is. And oftentimes the mutations for different cancer types are specific to that cancer. Or there's certain mutations that always occur in a certain type of cancer. So we know if we see a certain mutation, then we're like, oh, it must be this cancer, or it might be this cancer.
Oh, okay, that makes sense.
Mm-hmm.
So you're basically using a gene then, I assume that it's not one that codes for something that differs a lot from person to person, like hair color or eye color, but something that's common to all patients.
Yeah.
Excellent. Well, maybe we can go to some more general questions then, since we're a Cancer 101 talk today. What inspired you to pursue cancer research in general?
So I got interested in research pretty early on. So when I started my undergraduate studies at the University of Toronto, I started volunteering at a genetics lab, and just the kind of research they did was really cool because like you learn about a lot of things in textbooks and then you kind of see that happen in the laboratory. And then I continued to do some research over the summer, and then I did a thesis project in my 4th year of undergraduate studies, and that was in a cancer immunology lab. And I got to see a lot of the things that you hear about in the news regarding cancer research, and just firsthand. And I was like, this is really pretty cool stuff. And I was interested in it. I kind of just continued on that road and applied to graduate school, and here I am now.
Still on the way doing research as you go. Do you have an ultimate goal in mind for your career, or at this point, do you still have some branches you could choose between?
So the interesting thing is there's no one route you can take from where I am right now. There's so many opportunities out there. And at this point, I just want to kind of dip my feet into many of those opportunities and see kind of where my interests lie. I definitely know I want to stay in a clinical setting. So I want to be able to interact with the clinical side of things, so what you see in the hospital setting, as well as what happens on kind of the bench, so in the research field. So if I find something that I can integrate both parts of research and I guess clinical stuff into, then that would probably be my ideal pathway or route.
That's really interesting and good to hear that you are this far beyond the high school level, the undergrad level, you're in graduate school, and yet you still have multiple options for where you can go from here. You didn't have to figure it all out when you were in, grade 11 or 12.
No. It's way too early to stress out about that at grade 11.
So what is the most challenging part of being a researcher in your field?
Challenging. Honestly, it's keeping up with all the different fields. My research is very specific, and you think cancer research is like one topic. It's honestly not. There's so many different branches to cancer research, and if you really look at it from like a high level, all these different aspects of cancer research is integrated. So like keeping up with all the different branches is probably the hardest part because everything changes on a day-to-day basis. So we always find something new that we didn't know yesterday, and just keeping up with that is probably the hardest.
Well, and to be fair, I'll ask the flip side. What is one of the most rewarding parts of doing what you do?
The rewarding parts from a research perspective is when experiments go like you expect them to go. You can plan, and it might take you weeks and months to plan an experiment. Sometimes they may not go as you planned, but when it does work, it's the best.
So I think that's important for our audience to know too, that you do all of these experiments, but they don't work every time, and maybe not even half the time, depending on what you're doing.
I'd say 10%, maybe that's even being optimistic. 10% of the time is when it works.
So maybe something a little bit away from research now and more about cancer and lifestyle. What can our audience do? What can the students do to reduce the risk of cancer? What do we know so far?
So there's a lot of, part of cancer is definitely genetic based. but there's definitely an environmental aspect to it as well. So maintaining a healthy lifestyle is definitely key. And you hear a lot about this, but it's actually very true. Maintaining a good nutrition, exercise, wearing sunscreen, very important things. And these are just like some of the common things that you hear in the media or like even in school that, you know, people may not understand the importance of, but it definitely plays a role to some degree.
And do you have any knowledge of the efficacy or how well things work in terms of those really well marketed specific things, like a specific type of food to eat, or you know, doing something with lots of antioxidants because that's going to prevent cancer?
I can't speak too much about the specific marketed items, but I think just maintaining a good diet, obviously not eating a lot of junk food, because that definitely has bad influences not only on your health and lifestyle, but in terms of, you know, the environment for the organs that are digesting that food. You want to make sure that the organs are healthy, because if they're not, then they're probably going to become unhealthy. And it definitely has an effect at the organ level. And that's sometimes, particularly in colorectal cancer, poor diet is oftentimes one of the causes of it.
Well, it's good to hear that it's not overly complicated. It's all the things that you know, are best to do for ourselves for many reasons. They're also beneficial to reduce the risk of cancer, rather. So we have a little bit of time left, so I'll go to a few more questions. Maybe what types of other experts do you find yourself collaborating with in the course of your research?
So a very big field right now in research is genomics. So I talked a lot about DNA mutations. We now have the ability to sequence our DNA and really look at how our DNA has been coded. And this is a very, very big field, and it's advancing a lot as well. Today, we're able to take a sample of someone's blood or a skin biopsy, and we're able to sequence that entire person's genetic information, which is crazy. In my field, in my research, this is very important because blood biopsies or liquid biopsies or blood tests for early cancer detection relies on these mutations. So we need to know how the DNA in a tumor or how the DNA in the normal cell is coded in order for us to go into the blood and really pick out what we're trying to look for. So oftentimes in my lab, a large portion of our collaborations is in the genomics field.
And what is the thing about cancer, maybe cancer research, maybe cancer in general, that you have found to be the most surprising, or one of the things?
Surprising thing about cancer. So cancer, we do not fully understand it even today. I spoke about the genetics or the DNA level, the DNA aspects of cancer, but we have recently found that it goes beyond that. It's not just the way the DNA is coded, it's also about the way the DNA is modified. So if you can think about it kind of like a Christmas tree, the way the tree is decorated. In a sense, our DNA is decorated as well with different molecules. Recent research has shown that even if there may not be a mutation in the actual code to the DNA, if the DNA is not decorated as it should be, then that can also lead to cancerous events. And that was kind of interesting because you would think that the code is where it ends. The DNA code is the ultimate end, but it's not. There's definitely more to that, which was kind of surprising and interesting.
It sounds like even the researchers like yourself and the people you collaborate with who are sort of at the forefront of what is new and what we're just learning now, it seems like every time we get one step further, we realize there's another step and the staircase is even longer. Things seem very intricate. It's kind of an amazing thing to learn about. So I think at this point, we can wrap up. I know our guest expert here has many commitments in her day. Thank you so much for taking the time to let us know a bit about cancer, a bit about the research that you are doing, and help us to get a little more awareness before World Cancer Day.
Thank you, and you're very welcome. I was happy to share a little bit about the knowledge I have, and I hope you learned a little bit too.
And just before we sign off, I'll let everyone know that next week on PIR Live Event, we'll learn how math can be used to predict the spread of disease or help the NHL decide which city to expand to next. More information on this and other upcoming webinars is available at pirweb.org. Thanks so much for tuning in, and have a wonderful day, everyone. Bye-bye.